So I'm working for the CNRS, and for full disclosure, we started a company named Bionomics in 2020, just in the middle of the COVID, so it was fun. And I have a good news for you today, is that we're gonna talk about I apologize for the private joke for the one who was not there, but that's your slide, Roberto. Okay. Not exactly QTL. Technically, this is the plant of my talk. So I'm gonna give you a very general introduction to give you an idea of what we do in the lab and all the questions that we have. I was thinking about talking about how we use generative AI to control biological process. So that was planned. I'm going to talk about this. This is your part, Roberto. So we're going to talk about genetics. But in the middle and seeing all the presentation the past week, I also tried to stick a bunch of slides about what we do about plant nutrition, about nitrogen and phosphorus, basically. So about the general introduction, I'd like to point to these very nice pictures that you have on the workshop. But I think it's a bit depressing for one reason, is that in Western world, the time goes this way and it's not very fun, huh? Okay. So I guess what we try to think is that and this for an AI, this is Arabidopsis. Okay, I say plant Arabidopsis in the desert, but
don't forget to send me for the future.
I will send it to you.
Should put the week on it.
Yeah, we're gonna ask removing the health. How many dead? Okay.
They're all dead now.
Okay, they're all dead. So this is not exactly Arabidopsis, but it looks like it because
we hire twins, huh? Actually.
Yeah, that's a
clone of scientists. Once you find the right one.
One with glasses and not
they're all white, so that's
miracle
okay.
Okay, good. So then we hope that's gonna push a little bit more, and then hopefully we get this kind of stuff. So the context, like the biological context, is something we already heard about, but I want to bring to your attention that nitrogen is very important, and it's the Haber-Bosch process that just take the nitrogen from the atmosphere and make it available for plant and for food chain is a huge discovery, and some people consider that it saved more. It's the major discovery that saved more life all over the planet. And some people consider, and I think it's a general statement and people agree on that more than half of the population on the planet is supported by this chemical activity. But of course, it's a double-edged sword in the fact that, for instance, ammonia production through this process uses about 2% of the energy worldwide. And in Europe, this energy is mostly from Russian gas. So it also have like political consequences. And this not only use energy, it just releases CO2, and also the use of nitrogen itself release not only CO2, which is a very important climate change factor, but also it releases N2O, which is also a very important gas for climate change. So it's a very interesting aspect, and I just encourage you to go and see the context of this very excellent speaker, Christian Huguier. I don't know how you say it basically, but it was an amazing talk, and it's if it's recorded, I send this to you. In particular, I took a picture of this very interesting slide. It was a bit complicated because it's a 5-dimension slide, but the work that we do is here, basically in his talk, where he tried to push the use of nitrogen per hectare. And our activity is basically, and everything I'm going to show you in the next slides is related to this particular arrow in his talk. Anyway, so I like to start my talks with this particular video, which is like the best plant in the world, named Arabidopsis thaliana, in its natural habitat, which is a petri dish, growing probably more Arabidopsis are growing in agar plates than in nature, I think. I'm not sure about that, but And as you can see, it's been split, like the root has been split in two parts. On one side, we provide nitrogen, on the other side, we don't. And you see clearly that the plant is able to colonize the side of the plate where nitrogen is. And not only that, I show you later, but not only that, but also it's able to repress its growth on the side where no nitrogen is. So in some way, the plant has a capacity of adaptation of its development in order to forage for nitrogen. So if you could harness this kind of phenomenon, you can improve nitrogen use efficiency. Not only that, so as you see here, that's a different picture of the split root. Not only the development is increased by the nitrate, but also the capacity of the root to transport nitrate through transporters, dedicated transporters, is also controlled by the presence of nitrate and with systemic signals as well, with signals coming from the shoots. So our question in the lab is try to understand the molecular events that are behind this phenomenon in order to eventually control that and improve nitrogen use efficiency. So how do we do that? Most of the time, for most of the approach that we have to understand this, we do transcriptomics. And for a long time, we use a bunch of gene regulatory networks in order to understand which gene control which particular target involved in these processes, including nitrate transporters and other nitrogen-related genes. And we are very much interested in to understand the role of transcription factors in particular in this particular case. So to give you an idea of what we do in the lab, we have two groups within the lab. People doing molecular physiology and people doing like some mathematics and geekery, okay? The molecular physiology, one way to see it is that we are very much interested into understanding how nitrate control a bunch of genes and what are the genes involved in this nitrate signaling pathway. And some of the nitrate regulated genes not only belong to the pathway, but also can be very interesting plugs toward the control of the other processes. Like I'm going to show you that phosphorus, for instance, is very much controlled by nitrogen signaling in a few slides. So today, I'm going to give you a few words about this. And then the two last points of my talk are two examples of the geekery that we are doing right now. Okay, so that are the extra slides and probably the extra minutes I'm going to take on the 30 minutes that I have. Just to bring you to your attention, one of the results that could impact nitrogen use efficiency. So technically during a PhD thesis, we've been working on a bunch of transcription factors that are named HHOs that belong to the GARP family. And very long story short, we at one point, we figure out that these guys are very much involved in repressing nitrogen transport when the plant has satiety. So if you feed plants with nitrogen, at one point, it figures out that it has nitrogen and it doesn't want to have too much, so it just repress the uptake. And these guys are involved in that. And if you do a quadruple mutant or a sequential deletion of these different transcription factor, you see a de-repression of the nitrate transporters transcriptional activation. And at the same time, you see an increase in the transport capacity of the root in a very high affinity nitrate transport. And these mutants are somehow able to transport twice as fast the nitrate as compared to the wild types. And that correlates very nicely, at least in hydroponic conditions where the nitrogen is not limiting. It correlates very nicely with the speed of the growth. So in some ways, and we are trying to translate that into crops, in some ways, by messing up a bunch of transcription factors, you can also release the nitrogen nutrition a little bit by controlling this phenomenon. Another thing that we found serendipity driven, it was like just a mistake. So we were doing controls to understand if our plants were controlled by nitrogen and phosphorus, and we figure out that these are two phosphorus starvation genes. So if plants want phosphorus, they just light up these two guys. And then we were studying the role of nitrate for the control of the development of the plant, and we figure out that these two markers in particular are very highly controlled by phosphorus starvation. It's what you see here, several hundred folds. But when nitrate is missing, these plants are not activating the phosphorus starvation pathway. And again, a long story short, we've been working like a couple of years on that, and we found molecular actors and so on and so forth. We also found that this phenomenon is maintained in croplands such as rice and wheat with the collaboration with a bunch of friends. And very long story short, so we identified a bunch of molecular actors, and many people did since 2019. And so our contribution, we were the first one to see that, but now there are a bunch of people working on that. And then, as you can see now, there are very, very active discovery on the relationship between the phosphorus nutrition, nitrogen nutrition, and we believe that can be very helpful in understanding nitrogen use efficiency. Okay, so now let's dive into the part number 3. So this story starts just like a game that we had in the lab. So when the AlphaFold came out, of course, it's been a revolution. Everybody knows that. It's used primary sequence, a very complicated deep learning model named AlphaFold 2. Now we have the 3, predict the 3D shape of the proteins. And when this paper came out, just with a bunch of geeks in the lab, they say, okay, it would be super nice to do the other way around. Just take the 3D shape of the protein, mask the different amino acids, and ask if, knowing this position and the neighborhood of this particular amino acid, can you guess the amino acid that is missing here? So this is what the model is doing here. Our model named Petribert is doing, is like, you take a 3D shape and it's gonna invent in a primary sequence. Interestingly enough, with this kind of technique, and of course, we are not the only one to do that. There is like a very famous lab that does that. Their model is called protein NPNN. But technically what you do is that you can start with the original protein. This is the GFP. You can use AlphaFold to fold it, or you can even use the crystal structure of GFP. And then you can use reverse folding technique in order to provide synthetic proteins that are supposed to fold this way. So interestingly enough, when you compare the original protein and the synthetic one, you have very low identity and very low similarity found. So one thing that we asked at that time, it was like maybe 3 or 4 years ago, is that since these proteins never been invented by evolution, but it's predicted to fold in a good way, can you produce them in E. coli, for instance? So we did gene synthesis for 15 different proteins, and at least 13 of them was expressed at the good size and so on and so forth. And since it was GFP, our expectation was to have them glow, okay? And the bad joke on this slide is that this is the positive control, okay? Yeah. So I was chatting with a good friend of mine, very structural biologist, and he said, yeah, but that was a mistake to choose GFP. That was a dead end from the beginning of the project. I said, okay, thank you very much. And he said, yeah, because GFP is known that if you do 1 mutation, it can change color. If you do 2, it just stops glowing. So what do you expect if you change like 95% of the amino acids? I say, okay, that's true. And he said, you should have chosen a luciferase, because luciferase has been shown and known to have evolved in different scenarios, and the primary sequence of luciferases, even functional ones, are very distant, although they have like the same structure. So you should have used this. So it tells me that on the Friday, I go back to the lab and say the geeks, okay, we want to repeat that on the luciferase. On the Monday, this paper came out. So it was at the same time discouraging, but also very cool, because we didn't invest a single dime into the deal. And so it's been a total nightmare, but at the same time, we learned a lot of stuff. So I think that these AIs teach us a little thing about the relationship between the structure and the function. I think it's much more like a continuum, where some proteins have like a very strict relationship between the structure and the function, and the other ones may be more sloppy in this case. Anyway, so we ate the bullet, as Americans say, and we move forward a little bit. So I like this slide. I use it to sell my project to people who don't understand biology. What are the common points between these three scary guys? The common point is peptides, okay? Like these venoms, the things that makes you very, give you a lot of pain are peptides, and what kills you in alpha, in amanite is alpha amanitin, and it's also a peptide. So it's interesting to think that evolution is selecting peptides as biological hacking functions a lot of times. So as well, I've been working a long time with Jean-Philippe Cormier trying to find peptides traveling from shoot to roots in plants, because peptides are known to be reporter of nitrogen status of the plant. So I've been working with this guy who is very good at discovering new peptides for a long time. He's in Toulouse, and he also has a company that sells peptides that you can spray on plants to control flowering and so on. And of course, peptides are very important for many things, including like pollen tube growth. You know, this is the way pollen tube find the ovule is by a peptide attraction and so on. So it's a very important field of research. And in the context of the Petri-Bert and AlphaFold, the hypothesis is that the AI learned how to shape a protein from a primary sequence. So it's very likely that it learned the biophysical interactions between the different parts of the protein. So these interactions are very likely the same one that's gonna make a peptide bind or not bind. Okay, so we decided to develop a new approach that we call Petri-Bert 2.0 to design peptides basically. And then of course the idea was to do functional screening in plants to go very fast into the validation. So this project has started like a year ago, and this is the model that we developed. So technically you take a given target, you can choose the residues where you want to put a peptide on. You have you have three steps, A, B, and C. The first one uses a hallucination of diffusion model in AI in order to choose the polyglycine, just the form of the peptide that will sit somewhere. And then you have the reverse folding that can be Petri-Bert or other kind of models that are gonna choose the lateral chains in order to have the best fit of the peptide with the target. And then C is the filtering using AlphaFold multimer and so on. So technically we do like 10,000 of peptides, and we do a lot of filtering steps in order to find the best one. We order them and we treat plants and so on. Just to show you a diffusion on the go. So this is like a given target protein, and you see this is 5 different polyglycine, and this is different evolutionary step of the polypeptide, and you see at the end it chooses different way to fit the polyglycine, and then the next step will be to choose the best lateral chain of the amino acids in order to have the best fit. All right, so as a proof of concept, we started with luciferase this time. We learned the lesson. We developed a bunch of peptides, and you see we have several readouts, including foliar discs of plants expressing luciferase and the strong promoter. And you see if you treat with a bunch of peptides, with a control peptide that has nothing to see with this, we have a nice repression of the luciferase activity in leaf ticks, but also totally in direct regulation, where we order the luciferase from Promega and do this purely in vitro. And you see we can have effects as low as 10 to 5 nanomoles of the given peptide. Of course, we wanted to go to plants, and then we went there by choosing a good readout, but also it has political consequences because people ask scientists to get rid of the glyphosate at one point. So the glyphosate is the most used herbicide worldwide, and it kills the plants quite nicely, and we have resistant plants. So by trying to target the same protein, our readout was, okay, if we order a peptide, we put it on the plant, it grows or it dies, it's a good screen. So we started to do that. the protein is very well known. It's involved in amino acid biosynthesis, essential amino acid biosynthesis, and we exactly know where is the glyphosate binding and all the residue in the protein that are supposed to bind the glyphosate. So we aim at the same ones. And this is the kind of protein, the kind of peptide that we had that has been designed by our model. You're gonna see that also the lateral chain that has not been chosen by random. You clearly see here that you have like a positive charge amino acid that are facing negative charge amino acids. So we think it could be very interesting to have like a good fit. So very long story short again. So what we found, so this peptide, so we ordered like a dozen of them, and we tested them a lot. And this is the best one. We repeated the experiments something like maybe 10 times or 20 times. This is what we found on tobacco. So these are plants day 0, and these are the same plants at day 5. And what you can see here, we inject the peptide. We have a penetration problem so far that we can try to solve. And this is a bona fide control, which is the scramble peptide. And 5 days later, you see this plant just stop growing. Just you have necrosis on this leaf. And then interestingly as well, we have long distance effect of necrosis here as compared to the control. And it also works on Arabidopsis. You see the peptide 4 is here as compared to a peptide that does not work nicely as well. So we are moving forward into this. So last part for you, Roberto.
Thank you.
Perfectly in time. So it's not exactly QTRs, but you know what's the difference between a GWAS and a QTR? It's just the precision. Huh? So this is a project that was born a long time ago, again just by serendipity, and it was pushed out by the COVID. Because when you are during the COVID, we were stuck at home. So the only thing that we had to do is it was bioinformatics and computational science. So everybody knows what is GWAS. So just to be on the same page, the idea is to simply correlate the genotype variation in the genomes together with traits in humans or in plants. And this is the very important paper from the 15 years ago, one of the first GWAS in plants in Arabidopsis. So here you have the 5 chromosome, and each dot here is a particular SNP. And the higher the dot, the best statistical correlation between the variation in the SNP and the traits. Everybody knows that. In human genetics nowadays, we have more than 500,000 people being genotyped, even more now with 23andMe and these kind of companies. And for the SNPs, in average, you can retrieve up to 10 million SNPs, depending on how you make a SNP coding and so on and so forth. What is cool in Arabidopsis is that the gene density is so high that with something like 500,000 SNPs, you already have like a gene density, a gene resolution. So like a SNP will land close to a gene, very likely. One big problem with GWAS that emerged 10 years ago is that it's the problem of missing a heritability. So it's a complicated word just to say that if you take the top SNPs on a given GWAS and try to explain the phenotype or the variance explained by the top hits, that most of the time you explain, if you're lucky, something like 30, 40, maybe 50%. Yeah, if you're super lucky. No, you disagree?
If you're lucky.
If you are lucky. But most of the time it's between 15 and 30%. And it's true in human genetics as well. You have like a big chunk of the heritability that is not provided. And one explanation, among others, is epistasis, which is also the equivalent of saying, okay, it's probably the combinatorial game between the SNPs that makes, between the variants, could be something else than SNPs, but between the variants that can explain the phenotypes. So technically, this is a slide I use for the students, but if you have individuals having a T at this position, you see you're more likely to be sick. But these two individuals, 5 and 6, have a T and they are healthy. And that can be explained by another SNP somewhere in the genome that's going to correct for that. It's a combinatorial game. The big problem with that is that you have if you get into an exhaustive analysis of such a big combinatorial space, you're going to crash a computer because it was supposed to be impossible to solve. In other words, if you want to repeat that, it's just say you want to do the exhaustive combinatorial game, you have to subtract the effect of one SNP and repeat the GWAS. Extract the effect of number of SNPs, which is to the square divided by 2. So technically, it's very difficult to solve. So after a COVID pandemics and some thinking, so I'm going to skip this. No, I won't. I have time. So just to give you an idea of how the data set is managed. So what we have is that we have the genotype matrix that we call X, which is the individual per the number of SNPs, which is most of the time like 1 million per, like in plants, like thousands individuals. In human, it could be 100,000 individuals. And in fact, in front of this, you have a phenotype that we call Y, that is a vector, could be a matrix as well. And the regular model tries to explain the phenotype according to X, this matrix, loading θ1. θ1 is the effect for every single SNP, is the effect of one SNP, and then the effect of the other SNP. Basically, is the regular GWAS version. What we propose now is the model number 2, where we try to explain Y according to X and θ1, which is the effect of single SNPs, but also with the effect of Z, which is the matrix of the all combined SNPs 2 by 2. This is it. Like Z is X star X. If you take a SNP, you combine this SNP with this SNP, this one with this one, this one and this one. So it grows to the square. But to be honest, this matrix, if you go on real life with 100,000 of SNPs, it's a monster, because it ends up to have like 100 billion columns, which does not fit a hard drive. Of course, does not fit in a RAM, it does not fit in a computer. So you have also computational problems that you have to solve. Long story short, we're gonna look at θ1 and θ2 later on. And just to let you know that we kind of solved the problem. So the solution has been published like a few months ago now in Genome Biology. It's 20 pages of hardcore mathematics, but and I'm not able to understand it. I just give you the Reader Digest of it. It's all about compression. So it's a technique of compression that is named compressed sensing, that has been invented by Terence Tao. That is one of the most super big star in mathematics. So that we use for genetics. So this compressed sensing just takes the huge matrix, and Terence Tao shown that you can compress a problem in a small word following some using matricial compression, only if this data follows certain criteria, features that hopefully the genetic data follow. And this is something that we discuss in this paper. So what we do is that we compress the problem in a smaller word, and we also choose the level of compression in order to fit it in GPUs, like graphic processing units, which are these graphic cards used to do AI and stuff. So not only we fit the mathematics to comply with the machine, and then we can really accelerate the calculation. And then it's been shown that there is no loss of information, and then you can decompress the data perfectly. Okay, just to show you now some quality control for regular GWAS. So this is the mixed models, regular mixed models that are used by a big part of the community doing GWAS. And this is our technique that we call next-gen GWAS. You see, this is Arabidopsis GWAS result for bacterial disease resistance. And this is a peak that is very well known. because below this peak, there is like a very known gene for bacterial resistance. And you clearly see that we have a quite nice correlation, except that this takes few minutes to calculate, and this takes like few milliseconds, so a fraction of a second. What takes a long time here is just to load the data. Solving it is almost like a snap of a finger. What we have found as well is that this correlation is nice, but sometimes for some phenotypes like flowering, we don't find such a good fit. We have like the good congruence, but it's not a perfect fit. Interestingly, is that the FSC locus that sometimes disappear in GWAS studies, in our hands, it show up to be in the top 2 of the SNP as compared to the top 20 of regular modeling, like the regular mixed models. That means the way also the machine deals with the SNP correlation, that is an important thing to deal with, can be a bit different in our hands, and it can be helpful for some phenotypes. So now let's move to the actual cool stuff. So now we are able to do more than 500 full SNP full combinatorial analysis, which is about 100 billion interactions. Instead of computing this during years, it takes us around 2 hours to compute. On a regular server, it's not a supercomputer, it's a regular server that costs like 30K or something. Fun facts, because our brains are not plugged to just manage this level of data, this amount of data. Do you have any idea of how much is 400,000 seconds? You heard that already, so you cannot say that. Just I'll let you know, it's like 5 days, okay? And how long is 100 billion seconds? Roberto, how long?
I don't know.
It's around 3,000 years. Okay, so it just gives you an idea of the effect of squaring this data. So it's a huge amount of data, and the results, 100 billion data points is 1 terabyte of data for one phenotype. Okay. So of course, it's also a challenge to display this data. So the company that you have here developed this kind of tiling results. So this is an epistatic map now, where you have the SNPs on one of the 5 chromosomes here. This is Arabidopsis, the SNP of 5 chromosomes here. And it's a square matrix because you have all the combinations. And this displays 60 billion data points for the phosphorus content of Arabidopsis rosette. And then we did a tiling in order to display that as in a Google Earth, for instance. So here you see that a heat map corresponds to the theta I shown you, and then it's like a 2D Manhattan plot, and the strong signals you can follow, and you see the layers just move in. And when you reach layer 0, like one particular square here is the interaction with 2 actual SNPs, okay? So of course you can click on these guys. You can right away get the positions here. And also you can do haplogrouping. I'm gonna walk you through this. So these are the results that you can obtain. This is like a 2D full epistatic map of Arabidopsis. You have 2 very strong epistatic signals here, layer this one and this one. And let me walk you through this one. So this is phosphorus content. This is the phenotype on this axis, and this is the different haplogrouping. So two SNPs seems to interact in this location on chromosome 5 and chromosome 3. And you can totally see that in chromosome 3, Arabidopsis is fully homozygote, so it's only two versions. Whatever the version, the mean is not changed on this end. Here, same thing. Whatever the version, the mean is not changed. But when you combine these two, you have 4 conditions, and you clearly see that you have more phosphorus when you combine this condition and this condition here. And it's about a 15% increase in the phosphorus content of this plant. So, and this is a pure epistatic signals that can be totally missed with regular techniques. Okay, I'm going to go very quickly on this one. And I invite you to read the paper or come see me. The question is how much heritability we retrieve with this technique. The answer is most of the time we double it. It depends on the phenotype, but for most of the phenotype we've been working on, we can double the amount of genetic information that we retrieve from the 2D space as compared to the 1D space. Another thing for people working in humans or also for seed companies, we also try to predict the phenotype doing like pure prediction where we train on one set and we test on another set. Again, we increase, these are if every data point here is a different model and a different phenotype with different ways to do it. And you see most of the models lies above the diagonal, which is that when you provide the 2D signal here. And so we improve also the capacity to predict the phenotype very strongly. And we display here one good phenotype that we are able to predict, like molybdenum concentration in plants is something that we improved very much right here using 2D signals. Okay, now the company for Arabidopsis provide online software in order to start the project with us. And just to show you that we have things working now on human genetics in collaboration with people in Canada. So of course, our humans, we are fully heads. So this is it's an epistatic interaction between two positions, chromosome 3 and 10. Same here, you see this is 3 versions of the allele, mommy, daddy, and head, okay? And you see that the average of this phenotype, which is like lipid circulation in your bloodstream, we don't care actually, but you see that the average here is not changed. The average here at this position is not changed either. But when you combine the 3 and 3, you have 9 conditions. So let me show you DEF from here to here. You have an increase in the phenotype if you combine with the allele A. If you go DEF with allele B here, you have a flat line. If you go DEF with allele C here, you go decrease. So it's also a pure epistatic signal that you can find in heads people. So these are the people involved in the group. You can sort them based on their affinity with geekery or biology. Of course, we are collaborating with a lot of people. And thank you for your attention.